Okay, chapter 13 is about how you control microbial growth. In some cases, we're going to just inhibit their growth, and in some cases, we want to flat out kill them. This chapter is going to eventually lead us to antibiotics, but this chapter is not about those. This is more about disinfectants and antiseptics and things along those lines. So here's the very first thing you have to understand about this chapter. If I can get my stinking screen to move. There we go. Fomites. Fomites are inanimate objects that microbes like vegetative bacteria or viruses or fungal spores or even fungal cells can land upon, and then if somebody else touches that fomite that has that vegetative cell or fungal spore or whatever, they can pick that up and then become inoculated with that cell. So fomites can spread the transmission of, we'll call them germs, even though we don't like that term in microbiology. This chapter is about how we reduce the number of microbes on fomites, not necessarily in people, although we will again mention antiseptics as we go through this chapter. So the two factors that we consider as we're talking about either physical methods to control growth or chemical methods to control growth is, what is the object that we are trying to clean? Like, how clean does it have to be? As you look at the picture that your book shows you, you've got various objects from cars. Some of them should be cleaner than others. You touch the steering wheel all the time, and more than likely you touch your steering wheel and then you touch your eye, or you put your finger in your mouth or something. So your steering wheel ideally should be cleaner than the seat that your booty is sitting on. Pathogen resistance. If we're talking about food, there are specific pathogens that can be found in food. How resistant are those pathogens to the chemical that we are trying to clean? Versus if we're talking about sewage, that's a whole different class of pathogens. They are going to be resistant to a whole different set of disinfectants. So you have to be considering these things when you're looking at what chemical are we going to use to try to disinfect the object. So would silverware need to be sterile? No, you should not need to sterilize these items, assuming that you're using them for what they were intended for. They should just be cleaned or disinfected, which means we're going to reduce the number of microbes that are on those items. Now here's a trickier question. Would a scalpel need to be sterile? And more than likely, the second you see a scalpel, you think surgery, and so you're going to automatically try to think yes. However, it does depend on what you're using it for, because remember, you also use scalpels to dissect organisms in a lab. Well, you're not going to give your fetal pig an E. coli infection, so in that case, the scalpel doesn't have to be sterile. It should just ideally be sharp so you don't injure yourself in the process. So it really does depend on what you're using it for. That application part really comes into play there. One of the other things that comes up in this chapter is the biosafety levels. One of the things I wanted to address in here was what our labs are, and the Burleson campus, the Cleburne campus, the Hillsborough campus, they are all at BSL-2 labs, and that's because they have autoclaves. They have the air systems that can handle BSL-2 organisms. The organisms that we work with, as this chart says, they are indigenous to the area. We're not bringing in things that aren't from our area. Like we didn't bring a bat in from Africa just for fun to study it. And so we're at a BSL-2. We don't bring in other organisms. That's the aseptic technique comes up in this chapter. These are all the things that you do to try to maintain a sterile environment. As I say on this slide, that includes flaming your loop as you transfer from an inoculated broth to a sterile broth. You flame your loop after you're done with that so that you don't then contaminate the next sample you're going to work with. It includes, like you can see in this picture, the person didn't just take the lid off the petri dish and put it down on the table. You just tilt it up, because if you do take the lid and put it down, you've contaminated that lid. Also, by only cracking it, you're exposing to less air, so that's aseptic technique. Wearing the appropriate PPE, that means you're not going to be breathing on your samples, because then you're exposing them to more things that are coming from you. Gloves protect you, and it means you're not shedding bacteria from your skin onto the samples that you're working with. Keeping your hair tied back, same basic premise. And so all of that stuff helps you to maintain aseptic technique. Another important thing that comes up in this chapter is when you're working in medicine, you touch a whole bunch of different medical devices during the day. Some of them have to be sterile, some of them don't. And so there's actually a classification that tells you how clean each instrument has to be. Critical instruments are instruments that are essentially going to breach a barrier in the body. They are going to touch blood or body fluids. They are going to cut through things. So this would be needles, this would be scalpels. There's a clinical focus that goes through this chapter that talks about endoscopes. I personally believe that endoscopes should be sterile, and that's because if you're scoping a person, you're looking for something inside of them. You don't know what you're going to find. Maybe that person has ulcers. That means their surface barrier's been breached. They have an open wound. That means that endoscope should have been sterile when you put it in there. Some hospitals don't consider those to be critical instruments, and so they consider it to be a debate. It's really not, though, because you don't know what you're going to find. It should be considered a critical instrument, and that's something that you should fight for whenever you're going into a hospital. And if you're a patient, you want to make sure that that endoscope that's going into you, it should be sterile so that you don't get the carbapenem-resistant Enterobacter that the clinical focus talks about in this chapter. Semi-critical instruments, they are going to touch intact mucous membranes. So this is where textbooks put endoscopes, but based on what I just told you a moment ago, you don't really know if they're going to be intact when you're going in because you don't know what you're going to find when you get in there. Endotracheal tubes, those can often be the same thing, although generally speaking, the trachea doesn't tend to get as many wounds or ulcerations as the gut can have because of the just acidity of the stomach and whatnot, and so those are considered semi-critical instruments. Non-critical instruments, those are just going to touch intact skin. So the stethoscope that you can see in that picture, the gurney, the hospital gown, any of the leads that you're putting in for EKGs, things like that, those are non-critical. They don't have to be sterile. They should be clean, but they don't have to be sterile. What's the single most important thing you can do to protect yourself and to protect your patients? Y'all should know this based on what we've been talking about in lab from day one and just from living your life up to this point, and that's wash your hands. Washing your hands between patients helps you wash off any infection that the previous patient had before you get to the next patient, helps make sure that you are also not contaminating yourself in between. Or from touching that patient that maybe had that infection just a moment ago. So you should wash your hand after every single patient, and patients should really try to make sure you're washing your hands before you touch them as well. That does dry your hands, as we've probably discussed in lab, but they make lotion for a reason. Just moisturize as much as you can to prevent those cracks that can come in the skin. Wash your hands. There's no excuse not to. A question that pops up in here I want to make sure you know the answer to. So we always have you take your samples and put them over on the counter off to the side in the lab. We don't just throw them away, and that's because we need to sterilize them before we throw them away. The broths, the slants, the plates, all of those things, they are very contaminated, and we do not want to take a sample that has trillions of bacterial cells in them and just throw them in the trash, because then we are throwing a lot of vegetative cells out into the world, and we don't want to do that. Even if these organisms are not pathogenic, usually when you have them at high numbers like that, they can be. So we sterilize first, then we throw away. Okay, we have a whole bunch of physical methods that can destroy organisms or inhibit their growth. This is the table from the textbook that should help you fill in the table that I gave you in the notes. Make sure you follow the information that is in my presentation, because that is what I will test you on. It does pretty much follow what you see up here, but there's some stuff that I cut out. Like if I just put heat, I didn't separate all the different things like boiling, dry heat, oven, incineration, although I did have a separate line for autoclave and pasteurization. I just had pressure. I didn't have two separate lines for the different kinds of pressure, stuff like that. So follow the presentation that I gave you and you should be good there. Another question that pops up in the presentation is how do we sterilize our glassware? And it's with this device. You should recognize that from the lab. That is the autoclave. The autoclave, it can be sort of confusing because it does have high temperature and high pressure, but the pressure is not high enough to kill the cells themselves. The pressure is just there to increase the temperature of the steam. So it's there to increase the temperature. This is a temperature killing thing. How do we sterilize our inoculating loops? We incinerate. So that's a heat thing, a dry heat thing. Okay, I pulled this picture from a different textbook. The chemicals that are used to control microbial growth affect the cells in different ways, and this picture shows you some of them affect the cellular membrane or plasma membrane, some of them affect proteins in the cell, some of them affect the DNA. The ones that affect DNA, these happen to be the ones that tend to be pretty toxic to us because bacterial DNA is pretty similar to our DNA. The ones that affect proteins, those can be pretty toxic to us as well for the same reason. They're proteins built on the same structure as our proteins. These guys off over here, because they have a cell wall on top of it and their plasma membrane has some unique features, like they don't use cholesterol as a stabilizer and we do. These tend to be a little bit less toxic off over here. I'm not going to go through each of the chemicals. You can get that information from the full presentation that is posted online, but I did have this slide up here. So remind me why hydrogen peroxide bubbles when you put it on a wound. That's because of the catalase enzyme that you produce. Catalase splits the hydrogen peroxide. Some bacteria have that as well, and so that does reduce the efficacy of hydrogen peroxide for catalase positive organisms. The table at the back of the chapter for the chemical methods of control was not as copy-paste friendly for PowerPoint, so I pulled this from a different version, a different textbook. This textbook just called them biguanides instead of bisbiguanides, but same thing. I was going to see if there was anything else that I needed. Surfactants, they just called them the quats or quaternary ammonium compounds. That is a type of surfactant. So you can look here for the surfactant line. Ethylene oxide gas, that is an aldehyde. I didn't put that one in your notes because it's just an aldehyde, so I left that one alone. But there's that stuff. And then the clinical focus, I pretty much said everything I needed to say for that one. So I hope you learned some stuff about the importance of why we need to sterilize some things but not others. And we will get more into antibiotic resistance in the next chapter, which was what the woman was infected with. That is its own separate issue, but she wouldn't have had the infection had the endoscope been sterile to begin with, and that's why the disinfectant aspect was important for this particular chapter. Hope that helps you just as a summary for this chapter. Those two tables can be really helpful as you go through here. If you have any questions, as always, please don't hesitate to contact me.